So on to the main event for this evening, the Environmental Microbiology Lecture for 2022. As I've mentioned, the Environmental Microbiology Lecture is one of the most important events of our calendar, and we're delighted this year to award the Environmental Microbiology Lecture to Professor Kristala Prather, and we're delighted you have joined us this evening. Professor Prather is the Arthur D. Little Professor of Chemical Engineering at MIT, where she also is the Executive Officer of the Department for Chemical Engineering. Her research focuses on the themes of design and assembly of novel pathways for biological synthesis, enhancement of enzyme activity and biocatalysis, and control of metabolic flux, and bioprocess engineering and design. Professor Jones grew up in Longview, Texas, and was inspired early on by her physics and mathematics teacher to pursue a career in science, and what a career it has been. She gained a BSc, or Bachelor of Science, from MIT, and a PhD at the University of California in Berkeley, awarded in 1999. She then worked in bioprocess R&D and for the Merck & Co. company, the chemical company, in industry for 4 years before returning to academia and a faculty position in 2004 at the Massachusetts Institute of Technology, where she remains to this day. Professor Prather is well known for her trailblazing, and I say that as reading many articles about Professor Prather over the years. It's almost impossible not to see the word trailblazer appear in those documents. So your trailblazing scientific outputs, of course, have reached world recognition across the globe, but also your dedication to teaching, we spoke briefly about that earlier, and also your advocacy work. In your career, you have won numerous awards, many awards, and not least of those the Martin Luther King Jr. Leadership Award and a fellowship of the American Academy for the Advancement of Science. So, Kristala, on behalf of the Society, it's my honour and pleasure to invite you to give the 2022 Environmental Microbiology Lecture. Thank you.
You never know what people will find on the internet about you. I think that's the first time Longview, Texas has been mentioned in an introduction. I'm going to do my best to talk about what we're trying to do to address some of the issues that Professor Timmis talked about. I will just point out I have this note here because one of the pathways that I describe is the subject or the object of a license agreement for a startup company. But in the introduction, it was revealed that I'm a chemical engineer by training, and I always start here because I got my degree in the early 1990s, and at that point in chemical engineering education, this is all you needed to know about a microbe. Don't worry about what's happening inside of it. It was this thing, and much of the engineering education around biological systems came from the bioprocess perspective. So as chemical engineers, our job was to just figure out how the outside, how changing the environmental conditions would affect the productivity. But of course, what we all know is that cells are just collections of lots and lots of chemical reactions. And so this is a diagram from KEGG, the Kyoto Encyclopedia of Genes and Genomes. And just to orient you, if you haven't seen it, each of these dots or nodes represents a chemical species. And the lines between them are enzymes. They're the results of chemical reactions. And so as a chemical engineer, once I saw this, I said, of course, this is really just a massive collection of reactions in parallel and reactions in series. And so a lot of what I learned as a chemical engineer around how to optimize processes, we've been trying to apply those principles towards biology and using biology for the purpose of making chemicals. The challenge, of course, is that we want to start with something very simple, like a carbohydrate. We have to figure out how to navigate this very complex network to get towards a particular compound of interest. Along the way, we are well aware of the fact that there are so many of those nodes there, which means we get things that we don't want. So we have byproducts. And traditionally, within metabolic engineering, or within the scope of trying to engineer biological systems for chemical production, the solution to that is to say, well, any byproduct must be some dot within this matrix. That means that there's an enzyme that encodes, or rather a gene that encodes for an enzyme that produces that thing. And if I can just delete that enzyme, then I should be able to increase the productivity of what it is that I'm trying to make. So have more of what goes in going towards desired products. The problem, of course, is that we know biology, I'll say biology is selfish, right? Biology would prefer to take those substrates and use them to make more of themselves rather than the compounds that we're interested in. And so now you don't really have the option of deleting whatever the key enzyme is or the key gene for that enzyme to make your byproduct, because you need the cells to make the proteins that are the enzymes that make the things that you want. So it's this central challenge that we've been working with, which is really summarized here, that what we've been trying to do is to merge what we know of engineering design with what we understand of biology to build now new devices using the language of synthetic biology, but new control architectures that allow us to dynamically regulate flux within metabolic pathways, or to really be able to have the cells turn genes on or off in response to some environmental condition or some perturbation that we make. So we call these metabolite valves. That actually belies my training as a chemical engineer. I think about valves. If you are in the bathroom and you turn on the water faucet, that's a valve. And when you open it, the water flows. And when you close it, the water stops. So what we'd like to be able to do then is to have some branch point metabolite where when the valve is open, central metabolism is preferred. But then we have these genetic devices to close that valve and allow us to make more of the desired product. We first started working on this a long time ago, at a time when we didn't have as many of the tools available, but we were able to demonstrate the basic concept using a system where we could induce protein degradation. And simply put, what we did was to identify a gene that was essential for growth, and then in response to some perturbation, that gene would be targeted for, the enzyme rather, would be targeted for degradation, and that would restrict growth and allow more of the desired product to be produced. We were very focused on that time on using something that was independent of any particular pathway. So we are concerned about all these myriad of SDG problems that Professor Timmons talked about. And we think about being able to build solutions that are transferable amongst different cases and not necessarily limited to any one particular application. So we were able to use a small molecule to do this in order to trigger this degradation. Now, the problem was that this still required user intervention. And so this is now where my chemical engineer hat comes back on to say, if we want to be able to construct these biological systems that are more complex, ideally we want to be able to minimize the amount of interference within a manufacturing facility. So I'd like to be able to inoculate, have the cells grow, and make what I wanted to grow without someone constantly coming and checking. Okay? So we really wanted to go towards what we define as autonomous control, which means to endow the cells with the ability to know when is the time to have this particular switching mechanism. We use quorum sensing in order to do this, which we get from nature. So I will put in a plug for the critical role of environmental microbiology in helping us to understand what nature has given us as this ginormous toolbox to be able to pull from in order to now make purposeful changes in order to achieve these objectives that we have for greater sustainability. And just quickly, I'll point out that the way the quorum sensing system works, there are a few critical components. There's a quorum sensing molecule or a ligand, which is the product of a synthase, so an enzyme that makes that in particular. You then have a regulatory protein. It's shown here as ESA-R. And that works in can work in a number of different ways. It will interact or bind with the DNA, as regulators are known to do. And in this particular configuration for this system, binding of ESA-R in the absence of the regulator causes gene expression to be turned off. Binding of the regulator with ESA-R causes it to come away from the DNA. That leaves the promoter open. And so you go from an off to an on configuration, classic repressor mode of gene regulation. It's known as quorum sensing because there's a critical threshold that's needed of these signaling molecules to be able to trigger gene expression. And as you can imagine, if each cell has some of this synthase protein, it's making different amounts of that ligand or signaling molecule. And as the total concentration crosses some threshold or reaches some quorum, then you exceed that critical amount in order to trigger gene expression. So we were actually interested in this system for two reasons. One was because it had been studied by a colleague at the Rensselaer Polytechnic Institute where she had taken the ECR molecule and made a number of different mutants to give us a very wide operating range, but also because this system actually can serve as an activator in addition to being a repressor. And in the activating mode, it binds in the absence of the ligand as well, but in doing so, recruits RNA polymerase so that the default state is to be on. And then, as the quorum sensing molecule builds up, it still interacts with that same regulatory protein. The protein comes off of the DNA, and that then takes RNA polymerase with it, and the resulting gene expression state is being off. So we actually have some optionality here in terms of having something that goes from off to on, as well as from on to off. What we do in order to establish the tunability then is to build libraries where we have combinations of promoters and ribosome binding sites of different strengths so that we can span a range of expression levels, because it is the amount of this synthase which determines the amount of the signaling molecule, which determines the timing at which you would actually have this switching behavior. So from our library of about 30 different strains, we can scan a range of physiological states or physiological conditions for the cells to then tell us what is the right time period to have this switching behavior and maximize the productivity. So we can characterize our library by just using a reporter protein. This is a green fluorescent protein. It has a degradation tag because this is now testing in the on to off. And central dogma being what it is, if we stop at the level of transcription, but the protein is very long-lived, we can't actually measure that as a decrease or a change in any of the regulatory behavior. Instead, though, if we tag our reporter protein with something so that it will degrade, then we can see the emergence now of fluorescent signal, and then when we have the switching behavior go off, we can see a natural decline there. So in doing that, we could actually now, for this library of 30 different strains, test or scan this wide range of behavior. And what I have plotted here, this is ordered from the negative control to the positive control that doesn't turn off at all. And here's the OD, or the density at which we see this peak fluorescence, which we define as the switching OD. And you can see the library actually gives us a range of behaviors. There are two things to point out here that are interesting. One is that we actually have now gotten a lot further along this path of using real engineering principles as applied to biology. It took a while. There are a lot of wild claims being made about 20 years ago about all the things we're able to do, build a tree that's going to spontaneously turn into a bookcase. We're not there yet, right? But we do understand how to characterize promoters and ribosome binding sites. And so we actually chose these 30 because they were predicted to span a pretty wide range of behaviors. And so what's shown here is the predicted strength and what we actually measured at the switching OD, where the weaker those combinations are, the later you should see the switching behavior. And that's actually what you see in this particular case. But the other part of this, where we still have work to do as far as both the engineering tools and the biological understanding, is the fact that only the very lowest levels of expression gave us a graded response, meaning it gives us a couple of different options to choose between and actually see differences in behavior. And when I say lowest levels here, you can see this is benchmarked towards something that's defined as 100%. All of these here, there's 6 that we use commonly now, they are 5% or lower. And part of the challenge here is, again, if we look at what nature has given us, nature has a number of different ways of regulating physiology and regulating metabolic responses. And nature tends not to just have very, very strong promoters cranking things out all the time. And so if we're now trying to play around with naturally occurring systems and engineer them to be more responsive, we have to be a little bit more subtle and more nuanced than we're on the side where we're just trying to say go more and more, faster, faster. Well, with this library, though, we could actually test out and see how the system will behave. And so this is actually a pathway for the production of a compound called glucaric acid. It is the pathway that is currently licensed to a startup. But you can see that what we're able to do here is in E. coli put together a few different genes in order to have this being produced from glucose. And I can start from the back and work my way up. We actually started by identifying an enzyme from a soil bacterium. This is from a pseudomonad that can produce glucaric acid from glucaronic acid. It turns out that there are a number of eukaryotic proteins, including in mammalian systems, where you can produce glucaronic acid from myoinositol. And a professor, before we started this work at Michigan State, John Frost, had shown that you needed only one recombinant enzyme activity, this synthase, in E. coli in order to produce myoinositol from glucose, and that relies upon an endogenous phosphatase. I will say, because Professor Timmis brought it up in his talk, he mentioned eutrophication and the problems with excess phosphate release into natural waterways. The interest that is now driving our commercialization of this product is to replace phosphates, to be able, in water treatment solutions, to be able to reduce the number of phosphates or the amount of phosphates that are released into the environment, and the very serious environmental consequences that come along with that. So now back to the more academic interests. If you look at this, the first intermediate in this pathway is glucose 6-phosphate, and it's a really classic example of an intermediate that the cells really want to keep for their own purposes because it's central for primary metabolism, rather than having it be siphoned off into products that it doesn't really care about. So here's just a brief diagram of what central carbon metabolism looks like. So again, here's glucose 6-phosphate. You can see 3 different enzymes in E. coli that take that as a substrate. And it's these 2 that we're most familiar with. This goes to the pentose phosphate pathway. This is the Embden-Meyerhoff pathway. Phosphoglucomutase is essential but not sufficient. So you have to be able to make some amount of glucose 1-phosphate for membrane biosynthesis. So you can't knock this out, but you have to have 1 or 2 of these other 2 enzymes remaining to be able to grow solely on glucose as a carbon source. So I'll skip the details of how exactly we chose the target, but we ultimately decided to knock out glucose 6-phosphate dehydrogenase, so the ZWIF gene that goes into the pentose phosphate pathway, and we built our valve at phosphofructokinase, which is actually the first committed step, the first irreversible step in glycolysis. The idea, again, if we follow along with this theme of being able to use valves for control, is that when the valve is open, what you allow is for the cells to grow and to produce as much biomass as they want, and also, very importantly, to produce those enzymes that are going to do the catalysis that we're interested in. Then as a result of now the perturbation, this perturbation being scanning now, using quorum sensing, the right moment to have a switching behavior, we expect for this to be tuned down, and that would allow now a buildup of glucose 6-phosphate that gives us more of the first intermediate and ultimately more of the product that we're interested in. So we can take advantage of that quorum sensing system I described previously. The ESA-S variant of this promoter is the one that starts as an activator. So we start in an on state where we have high flux through here, and then in a certain period of time, we expect the switching behavior to downregulate expression at that node and then allow us to produce more of the product of interest. Again, I'm going to skip over a lot of details and just present you with one summary slide that sort of shows both how the system works and how we think about scalability. So again, I'm a chemical engineer by training, which means we like to play with cells, but we ultimately want to believe that we can put them into very large bioreactors and then produce something useful at very large scale. Unfortunately, you can't really do development at this scale or larger scales because you're limited both in access to equipment and in the cost of raw materials. And so we tend to do a lot of our development work in shake flasks. This is a classic 250 milliliter shake flask with a 50 milliliter working volume. So using that as a basis, what I have shown here are 3 of our different quorum sensing strains, where they are ordered from strains that switch very early to strains that switch very late. And what's shown in the 2 different bars are glucaric acid, which is our target compound in blue, and then acetate is an unwanted byproduct. And I'll just say very briefly, we've looked at this pathway for a long time, and in certain conditions we would see acetate built up, but not always. And so we were a little bit surprised when we saw that our wild type strain could only make acetate and actually couldn't successfully siphon off any of that glucose 6-phosphate into our product of interest. Whereas we had one optimally switching strain that had the exact opposite profile. It only made glucaric acid, it made no acetate. For a strain that switches very late, it looks like wild type because wild type doesn't switch at all, and so that actually has, again, a buildup of acetate, but doesn't have very much in the way of glucaric acid. And then a strain that switches early does a little bit of both. It can make product, but it also builds up a lot of this acid. So this was our baseline case. I talked about the large-scale reactor. The other way to think about this is in scale-down models. And so we also try to do as much as we can under smaller scale where we have higher throughput. So we wanted to see if in this configuration, this is now a 1 milliliter working volume compared to 50 milliliters, do we actually see the same behavior? And we do. So the system is very consistent in the sense that we now consistently use the bioreactor, as it's called, as our standard way of doing designs and analysis. And then we only scale up when we need additional material. That brings us now to the bioreactor. And the student who was working on this, when we got to this part, actually said, I'm not sure we should do that experiment. And I said, what do you mean? And he says, well, I think what's happening here is that in a shake flask, it's a closed system. We don't have any control of pH. It's a true fact, can't argue with that. As you might imagine, then if you make a lot of acetate, the cultures become very acidic. So the pH starts to drop very low. And so he said, I think the challenge here is not that these cells can't make product, but that the pH has dropped so low that they're no longer efficiently taking in glucose, and that's what we're seeing. And I said, okay. Keep going. And he says, well, if we go into the reactors have pH control. Yes, this is why we built reactors. Well, I think if the pH control is there, the results aren't going to look as good, so I don't think we should do the experiment. Okay, so those of you who educate graduate students, this is when you have the conversation with them that there's no such thing as good data or bad data. All data are just data. Your reaction to the data may be good. Your reaction to the data may be bad, but your job is to seek truth, right? And so go and do the experiment. And it turns out he was a little bit right, but he was a little bit wrong. What happened, and by the way, when we scale up to the reactor, we can't really run so many replicates, and so we only took the best performing case here. And so in our wild type strain, what we saw is that it did make a lot of acetate, but because the pH was kept neutral, those cells could reassimilate that acetate, which E. coli is known to do. And so it did make product. It made product later than the others, but it actually had the wrong balance in the sense that it mostly made cells and made a smaller amount of product. Whereas our strain that had performed very well in shake flask and in the bioreactor continued to perform well in the benchtop reactor. So I'm going to conclude this story here with a couple of things. One is that we were able to show that this was consistent across scales. And the other I borrow from my dear friend and colleague Tony Sinsky, who's a biology professor at MIT, who says the best metabolic engineering projects are ones where you start from nothing, which is this, and you make anything, because now you get to divide by 0 and say that you have infinitely improved upon your system. Okay? But the point here is that we now have this way of thinking about optimizing the systems that we're interested in, and we chose in this case a system that was independent of the particular pathway, and we've gone on to show that this is useful in a number of different regards. While that doesn't mean that you can't also think about this idea around autonomous control or dynamic regulation and apply it in a very pathway-specific manner. And so I'm going to do my last 2 examples on that. This is the same glucaric acid pathway where we had 3 cloned enzymes or 3 recombinant enzymes that we needed to make the pathway. And the second enzyme is the most interesting enzyme that we've worked with. And interesting, if you're a professor, is great because you can say, try all these things. Interesting, if you're a student, is often terrifying because this has given us many, many, many different challenges over the years. And one of the things that's very, very bizarre about it is that this is an enzyme whose activity is highest when it's actually cultured, or when the cells that produce it are cultured in the presence of its substrate. So if you're producing enzyme, if the substrate is present, you get much more active enzyme than if the substrate is not present. And we could actually sort of, it's also unstable, so we see high activity and then it goes down over time. And so if you think about that, that actually argues for the fact that you should wait and produce that second enzyme after the first enzyme has worked long enough to allow for some of the substrate to accumulate. And you can do this by actually just having 2 different inducible promoters where you are now regulating the time at which the second gene is being induced. And that's what's shown here. So this stops now at 2 steps in the pathway instead of 3 steps. And it shows that the later that you induce this enzyme, the higher your product titer is. So that's all fine and good, but that's not fitting in within our theme of having the cells actually understand and decide when changes need to be made. And I actually talked about this problem years and years ago at a conference in Germany, and someone in the audience came up to me afterwards, Julia Frunsky, and says, would it help if you had a transcription factor that responded to myo-inositol. And I said, why, yes, it would. And she sent us a paper before it had ever even been published, where they had identified this protein called iPSA that has a known binding site that they had identified. In the absence of myo-inositol, it will bind to DNA. In the presence of myo-inositol, it will come off of the DNA and allow the promoter to be open, which means we can use this to now have the substrate controlling when the enzyme is being produced. And we can first demonstrate that this works by feeding different amounts of myo-inositol with the reporter protein, and then showing that you get both increases in GFP as a function of increasing myo-inositol, and then what's shown here are different promoter strengths for that repressor. So the stronger the promoter is, the more of the substrate it takes in order for you to dislodge it. We could also then use that to show that if we were using this regulation to control the myox enzyme, relative to not having that point of control, we actually got substantially higher product titers. So we showed that driving expression of this protein from its substrate actually gave us increased production. And then, of course, the nice thing about it is that this control point of the cells versus the control point of the pathway are independent and complementary to one another. So we actually showed that individually, each of them gave us about a 2 to 3-fold increase in productivity, but putting them together gave us a 10-fold increase in the productivity. And it was the highest that we've been able to find in this particular strain of E. coli that had been known to be particularly difficult to produce this particular product. So I want to carry this theme through to the last part of the talk and talking about now the idea of using the substrates of a pathway to control how that pathway is being expressed. So I'm introducing a different target molecule now. This is glycerate or glyceric acid. It is used in polymers. It can be converted to any number of monomers to be used in polymers. It's used in surfactant synthesis. I'll say, quite frankly, it was just a very interesting substrate for us to work with, or product for us to work with, because we were very interested in how we could use galactoronic acid as a substrate. And I'll come back to that in a couple of slides to tell you about that interest. Okay, so this is a pathway now that actually still uses one of the enzymes that we had in our previous pathway. UDH and GLI are both enzymes added into the cells. That results in galactorate or galacteric acid being produced. And then these are endogenous to E. coli, where if you get this far, there are enzymes naturally present that will produce glycerate or glyceric acid. So what we wanted to do was to now have expression of these two genes actually be controlled by this substrate. And in order to do that, we identified a transcription factor from bacillus, so a different organism, and use much of the same process that we had before in terms of making the iPSA responsive biosensor. The different configurations here are just to indicate positioning of the binding sites of the operators, whether or not you have one site or two sites, and where those sites are located. What you can just take home from this is the fact that you do get a range of behaviors, both in terms of the dynamic range and the leakiness based on the configuration, so the exact sequence of nucleic acids and how you compose them. We were our primary driver here is to have something that both had high dynamic range, but that really had low leakiness as well. So this is the particular promoter configuration that I'll show you in the following slides. And the reason we became very interested in having some way of controlling this system is that it turns out if you're going to use galactoronic acid and make glycerate, you do need to control when these genes are turned on. Otherwise, the cells don't grow very well at all. So I'll just show you here. The gray is a case where we actually have the same promoter as in all the other ones, but that's one that's completely unregulated, meaning those genes are turned on all the time. And this is just a growth profile. So OD is a function of time. You can see these cells grow very, very slowly. What's shown in the other set in the pink now are cases where we used IPTG, so just a inducer that you could use for anything. It's not specific to galactoronic acid, and you can see how the cells grow there. And then here in this green one and in that dark blue one, green is where we have galactoronic acid control but no gal being added. Again, the cells grow fine. And then in this case, we add galactoronic acid, and you can see the cells are still growing very, very well. So we do need some kind of control in order to have a healthy cell. And then the question is, can we get away with not having to add anything extra and only adding galactoronic acid to the system to begin with? course, the answer is yes, otherwise I wouldn't be talking about it. But here are the data. We just published this last year, where this is now looking at substrate utilization if we have no IPTG or IPTG being added. And you can see those look very similar to each other. IPTG added initially, so at the very beginning where the cells aren't growing so well, versus 3 hours into the production. This is where no galactoronic acid has been added, so of course none is being consumed. And then here's our system that's being regulated, and you can see that it does a great job of consuming galactoronic acid. The cells that didn't grow very well actually didn't consume very much of the substrate and correspondingly made very little product. Whereas these were the IPTG control cases. Here's now the case of, again, no control, and here's the case of our substrate control. So what we've been able to find is that we can actually construct a system taking advantage of galactoronic acid as a substrate and not have to add anything else to it in order to get this product. And I don't have the yield numbers shown here, but we can get up to very close to 100% yield conversion on a molar basis of galactoronic acid to our product of interest. So now let me tell you why we care about galactoronic acid. And this comes back to the theme of the lecture, which is around environmental microbiology. So one of the things that we've become very interested in terms of how microbiology can make a difference is how do you utilize waste? We are very good, especially in the developed world, of taking more than we need, and the stuff that we have left over, we just throw it away. And mostly we ignore what happens to it. But a lot of what's thrown away is very rich in organic matter and is really ripe, we believe, for being able to valorize in some way. And that could be to be able to convert it into energy. It could be to convert it into additional feedstocks as well. This was originally motivated by a research grant that we had from the U.S. Department of Defense through the Army Research Office, and they were specifically concerned about food waste. That on their Army basis, they generate a tremendous amount of food waste every year. It goes nowhere, essentially, in terms of being able to use it for anything of value. The problem, of course, you have with food waste is that what you have as waste on Monday is different than what you have from waste on Thursday, because you serve something different on the menu on Monday versus on Thursday. So this is a very complex mixture that's going to change from day to day. And the goal is to be able to have some specific amount of one particular thing. So we really got interested in this idea of whether or not we could engineer a system where the output was stable, irrespective of heterogeneity in the input. So whether or not it was pasta on Monday or hamburgers on Monday versus pasta on Thursday or vegetarian lasagna, could we always get the same thing coming out? So from that perspective, it turns out galactoronic acid is is present in very high fractions in food waste, at least the food waste that we had information on. And gluconic acid, or gluconate, is another compound that would show up very frequently in these waste streams. So we wanted to sort of have this idea of having two different substrates, two different pathways that allowed us to take those substrates and make the same product, which meant that we needed to have ways to control pathways based on the presence of either of those two substrates. So this one, the galactoronic sensor I've shown you before, that's the EXUR that comes from Bacillus. We found a transcription factor also from Bacillus that responds to gluconic acid. And so what's shown here now is the response of this sensor to galactoronic acid, but also in the presence of gluconic acid, and then the corresponding data shown there to demonstrate that they are orthogonal to each other, meaning that galactoronic acid-based sensor will only respond to that, and the gluconic acid-based sensor will only respond to that one. You can see a bit of a decrease in the signal when gluconic acid is this high. We actually determined this to be a pH effect. So when you have that much gluconate in the system, the cells will actually consume it, but they secrete a tremendous amount of acetate. And we have later studies that show that we can correct for that. So we actually ended up with a different pathway and looking at same target, glycerate, but started from galactoronate here and chose to rely first upon the endogenous pathways in E. coli and then add two recombinant enzymes to be able to make the final product of interest. We had not done this pathway before, so we did have to do some optimization here, and I'll just show you really one summary graph that shows that. The X's here are knocking out all of these various competing enzymes that we know will consume our precursors, or our intermediates rather, for the final subject of the final product that we want. And you can see our roughly best performing case here is with this 6 sets of knockouts. If we knock out one more, these two aren't statistically significant in terms of the difference. And it turns out that knocking out this enzyme was especially detrimental when we added in a second pathway. So we actually started from this point here and then looked at trying to optimize different ways that we know how to put pathways together for higher production. In particular, we looked at increasing ribosome binding site strengths, and you can see we got an additional jump here. And then we actually looked at the ordering of these two enzymes, and we're actually able to find that if we swapped these so that the weaker enzyme was expressed first in the pathway, well known that in operons, the first enzyme has higher expression than the second enzyme, then that gave us the best performing behavior here as well. The reason we switched now from the original galactoronic acid pathway to this one is because it actually had a common intermediate that allowed us to use gluconate as well. So we actually have a downstream pathway that's in common, but upstream pathways that are specific to those individual metabolites. And again, here it's a matter of trying to knock out those genes that will compete and consume our substrate. We took our best case here. Looked at additional knockouts, didn't really make any difference, but if we improved the ribosome binding site strength from one of our genes, that gave us an additional benefit. And then it actually turned out that increasing the copy number to boost up even further the expression level of these genes gave us the highest production there. So we could put those two together, and something very interesting happened here that I will say in advance we don't know how to explain, and that is that if you start now with a single plasmid system, the best case I showed you of glycerate from galactoronic acid was about 2.5 grams per liter. It's only about 1.3 grams per liter from just having the pathway for gluconic acid. If we actually now have both of these in the same cell, but only one or the other substrate, it turns out this one goes down and that one goes up so that they start to meet in the middle. Again, I don't know why, right? And as we actually now change the ratio here, the differences in these are actually not statistically significant. So for reasons that I don't understand, we set out with this goal of saying, can we actually have something where the input is going to vary a lot and the output is consistent? And we sort of stumbled upon it by accident. So we're still trying to understand why. We also want to understand how extensible this is. So if we add additional routes from additional substrates, will we still see things leveling off, or we start to see some significant differences in how they're used? Reasonably consistent output from variable inputs. So I am right on time for the 40 minutes. I just want to summarize what I talked about today. The first one is that when we think about using biological systems for chemical synthesis, the first point of attack for optimization is just to figure out what are the byproducts. Let me knock out those genes that are contributing to that, or what's not being expressed enough, and really sort of push that as far as possible. And that's worked in a number of cases, but it also has not worked in many cases. And so we really think that having these other tools where you can look at dynamic regulation of metabolic flux just adds to the arsenal that we have available to really address these key sustainability problems. We've also gotten very excited about using metabolite-responsive biosensors from a particular perspective of addressing an environmental challenge, and that is to be able to contribute. I won't say we quite have a full circle, but at least have a longer arc towards the circular economy. So being able to take things that are now considered waste and repurpose and reutilize those in ways that it actually will benefit human society. I just want to end by thanking the folks who did the work. I'm highlighting Cynthia here because hers is the only work I talked about that's not been published. I have a draft of the manuscript I have to read on the flight back tomorrow. And the current group here who's continuing to explore these avenues that I talked about. I have our funding sources listed here. I really very much appreciate the invitation to be here and to share the stage with Professor Temis especially, and I'd be delighted to follow up with our discussion.